Okay, the following paper is called Cell Pattern Disordering during Convergent Extension in Drosophila. So I know there are many things here that it sounds confusing, like what is convergent extension, why they are talking about order and disorder. So I'm going to go through all of those. Yeah, and these slides were taken from a presentation of Rachel Bertel. And here, what I wanted to show you is that you can go from an organized state to a chaotic state. Yeah. So here you see organization because each one of these dots are having a similar arrangement. While here you cannot predict how the cells are these dots are interacting with each other. And what I'm going to show you is that in epithelia, we're going to go in an intermediate stage. Yeah. Well, it's not completely organized and boring, but it's not also so disorganized. Yeah. So you will see that when you see the geometry of tissues, they are slightly disorganized. Yeah. Because of the cell movement. Yeah. So when cells epithelium move, they have to disorganize a little bit to be as a result of the movement, as a result of the tension along the tissue. Okay, so let me show you what this group found. So here I'm going to summarize the key idea of this paper that I hope that you understand it now. There is a process called convergent extension that is found in different organisms, yeah, in early embryogenesis. So here what you see is an schematic of a Drosophila embryo, and here if you see they made lines of different colors, and when they label it, something similar to of what we have done, you see Drosophila legs. If you see the tissue got longer, yeah, got longer and narrower, yeah. So if you see now, you find that they start mixing it. The cells start mixing it, and instead of having 3 rows, you start having 2 or 3 rows, and it's different, definitely longer. Yeah. So this process is called convergent extension, and I'll show you this in Drosophila. It has been studied in many different organisms. And what this paper is talking about is studying this process of convergent extension in flies, and is finding that the cells in the beginning, they are not completely honeycomb pattern. Yeah, they are in between stage. They are not only also these disorganized pattern that you find in the soap forms. They are in between, and the reorganization, the convergent extension, it is going to disorganize the system. So if you put here a scale of order, here you have the highest order, it will be the honeycomb pattern, and the lowest order will be the soap forms. Here, the reorganization of this tissue is a process in between. Okay, so the hypothesis that they had in this paper is that the cell pattern in embryonic Drosophila will become more disordered during the process of convergent extension. So when they start, when the epithelial start moving, yeah, and they start interchanging neighbors, they are going to go from an organized to least organized. Yeah, so here you have a predictable lines here. With this schematic, what I wanted to show you is that it become a little bit more unpredictable, not as an unpredictable, yeah, not as disorganized as the chaotic, as the soap forms, but it's becoming more disorganized. So how we are going to test that? I'm going to show you that there is a mechanism that it has been studied for a long time. There is a way that we quantify organization in terms of numbers of cells, yeah, that it will give us the geometry of the tissue. Before that, I'm going to tell you a little bit about Drosophila embryonic development. So in Drosophila is a super interesting process in which you can divide it in 17 stages. So here you see the number, you see the different stages, and if you study, we spent one entire class talking about this in developmental biology. The coolest thing of Drosophila, or the unexpected things that I always repeat, is that while in humans you start dividing the nucleus and the cytoplasm at the same time. So you start having, every time that there is cell division in the embryo, you start with one cell, then two cells, four cells, eight cells, and you continue like that. In Drosophila, there is an early cleavage of the nucleus, but not of the cytoplasm. So you start having a cell that have multiple nucleus, yeah, and later the nucleus start migrating into the periphery, yeah, and then the cells are formed, yeah, the members are formed. So different from flies, sorry, from humans. Here, and I'm in the elemental biology, we will show you that there are many examples that are far from all the pretty examples that we find in humans. Uh, Here, what happened is that there is a syncytium, so one cell with multiple nucleus, and later the cell membrane appear when the nucleus are here. And here you have all the yolk, the food for the cells, yeah, and you have all the cells outside in the periphery. Later, after cellularization, there is a process called gastrulation, and during gastrulation, what you're forming is the three germ layers that are the ones that are forming the ectoderm, endoderm, and mesoderm, that are the three main germinative, the three main tissues, yeah, that if you take developmental biology, we will talk about that. And here there is a germ band elongation. So if you see here, the germ bands start getting longer. There you start forming all these parasegments. Then the germ band retract. Then you see this structure is going to disappear. It's called the dorsal closure. This tissue is called the aminoserosa. You need to memorize it. This is a wonderful example, the aminoserosa and the dorsal closure, to study tissue death, because all that tissue is going to disappear and is going to seal here like a zipper. You see here, and here is when the larval stages start. Um, do you have to memorize all these things? No, you just have to tell and remember that embryogenesis. There are many things happening. Yeah, there are a lot of movement. There are a lot of changes, and here they're just going to talk about this germ band elongation, this process that is very interesting, and it has been studied in a lot of detail, because it's an example of how tissues get narrow and elongate. That this is what happened, for example, for the formation of the spinal cord, for all those tissues, that the way that they form tubes in invertebrates. Convergent extension is important. All these long tissues require convergent extension, and what it has been found is that there are hundreds of ways to elongate a tissue. Yeah, and so this is a one example that people have studied in a lot of detail. Okay, so what they are going to do here in this paper is trying to use, as usually, describe the wild type. Yeah, that is the normal process of convergent extension that you can see here. And later they will study mutations. Okay, so they are going to study mutations, and in the normal wild type, you will have normal convergent extension, while in the mutant, you are going to have a disruption in the convergent extension. So look what happened during convergent extension. The tissues start getting longer and narrow. Yeah, sure. And when you have a mutation, this is not going to elongate anymore. So in other words, in convergent extension, you have these four rows of cells in the wild type. 1, 2, 3. This is just it's not that you have four rows, it's just a schematic to make life simpler. And here they converge into two. Yeah, now into two, and this got much longer. Yeah. When you have a mutation, and there are multiple mutations, you keep 1, 2, 3, and 4 rows, and later maybe they could be changes in cell shape, they got longer, but there is not actually the process of mixing of the cell, of the intercalation of the cell. This kind of things doesn't happen. So we're going to study we have a process that has been studied in a lot of detail. We have an epithelia, and we have many mutants. So this give us a lot of tools to try to understand how the order and disorder during this type of process. Okay, so how they did these experiments? They took confocal images. Remember, a confocal microscope is a microscope that allows you to take pictures uh, that have a high resolution, that allow you to remove the background, the background, the noise, and so they have high quality, and in addition to that, they can form three-dimensional pictures. Okay, so here we only have, I guess that these are only 2D pictures, maybe with a fluorescent protein of the boundaries of the cells, and this here you have A here, B, C, and D. So A is a wild type in the stage 6, B is a wild type in the stage 8. So if you see in this stage, it look more like a honeycomb pattern. This one, the geometry, it start changing a little bit. And the C and D, this is a mutation, and uh, that is of those of two. So these are two mutations that they are comparing what is happening in those two last stages. Yeah, so this is the type of data that they are getting to quantify the changes in the geometry, to quantify the the organization of the system. Okay, so how they are doing it? Super easy. So here we are going to have honeycomb pattern in this figure, and look what they are doing. They are studying how many neighbors, how many sites they have. So let's take one. Here they have in this cell, this one has 1, 2, 3, 4, 5, and 6 neighbors. Oops, 6 here. Yeah, so look, they have in the honeycomb pattern here, all the cells we're going to have 100% of them are going to have 6 cell neighbors, and you can do it later if you want. You can delete this one, and you can imagine we quantified, I think we did this one, we can quantify this one too, and start quantifying 1, 2, 3, 4, 5, and 6. Yeah, and you say okay, so you delete it again as I did it, and you can keep quantifying and see that all of them have 6 neighbors. When you have the wild type, yeah, you don't have all of them. They don't show a honeycomb pattern. Yeah? Only 60%, and the other ones are pentagons, and the other ones are heptagons. Okay, and while the tissue is reorganizing in the stage 8, do you see that there is a reduction in the order? So here you have less a lower number of 6 neighbors, yeah, so less hexagons, and you start getting also some an increased number of pentagon and heptagons, and you get even sometimes squares and octagons. Okay, so what you are seeing, what they describe in the system is that imagine those cells, the epithelial cells that we have been describing. Imagine that is Google Map. In Google Map, you see all the streets and houses, yeah, from the above, and what you see is houses, and you can see the ceilings, yeah. So what here the figures that these pictures are like the ceilings of the houses, and we see that some houses have ceilings that are squares, other ones that are pentagon. This is what we are quantifying here. So what they are saying is that if you see the cells from the top, they start changing in geometry, and they are going from a stage, the degree of order start reducing because of all the forces that are required to reorganize the entire tissue. However, if you compare with a foam, with a soap foam, you see that even there is a lot of variation. Although six, the hexagon seems to be important. Here you have a little bit more of pentagons, and you have even 3 and 9 neighbors. Okay, so again, honeycomb pattern the most organized, foam, soap foam more disorganized, if we think in terms of numbers of neighbors, and tissues are not so organized or not so disorganized compared to this. But however, the movement allows them to be more disorganized in this case. Okay, so I show you only one figure of this paper to show you the main idea of the paper, that they talk about organizational reorganization. And I show you that they do it by quantifying the number of neighbors. There are multiple ways that they can do that, and they show this quantification using mutations to evaluate how the process of convergent extension can change. But the key idea was that. So what is important of this? Because this process of convergent extension is very important, yeah, and it's found in very morphogenetic process. And it's important. You will see many papers that talk about convergence extension in terms of intercellular signaling, and is, as you see, there were 17 stages, I think, of or in the beginning that I showed you of the development of a fruit fly. So we are helping us to understand how this one work, can help us to understand how all of them can work together. So, conclusion: the hypothesis was proven to quantitative experiments. Yeah, and if you're watching this, you might be doing a project like similar to this one. Yeah, and we are trying to see how similar as the sexcom moves. Yeah, it reorganizes entire tissue. We want to know if we find the same patterns, that as a result of the rotation, the process it can cause a reorganization. However, during the com, there is much more things happening at the same time. So we just wanted to know what happened. Okay, so the conclusion is that uh, uh, these reorganizations are very important and show a lot of complex behaviors. And 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 we just show an example in which this reorganization of mechanical forces reduce the order of the tissues. And this is important. Why? Because it has been shown in other studies that when you reduce the order, so for example, if you take cancer cells and you try to see what happened during the if they have a honeycomb pattern or they show uh, something similar to the wild type or they show something similar to a mutant, they actually show a more disorganized stage. Yeah, so it's very interesting because it tends to say like what is showing this is that normal conditions, the epithelium seems to be organized a little bit. However, it can disorganize, but later it might come back to a normal stage. However, in cancer cells, it seems that the epithelium start disorganizing, and this can be a landmark of an early transition from a normal to a cancerous epithelium. Okay, I know that this paper has a lot of things. This was just the introduction. I hope that this was clear. And if you have any question, please let me know. Bye.